Okay, thank you very much. I'm pleased to be here to demonstrate to you guys how we're starting to use machine learning to answer some key biological questions regarding the genetic basis of stomatal density in C4 crop species, which is work I've been doing with Kevin, who's also in the audience today, and we're both in Andrew Leakey's lab here at the University of Illinois. So why stomata? So I'm sure many of you already know stomata are the microscopic pores on the lying primarily on the abaxal side of leaves, and they're the primary site of exchange of CO2 and H2O. Therefore, they're a key target for improving the efficiency of photosynthesis and water use. They're also implicated in the response to abiotic and biotic stresses. And crucially for us, the genetic control of stomatal patterning and density hasn't been explored in any great deal in C4 grass species. So we're trying to understand some of the genes that are involved in this in sorghum, maize, and the model species Soteria. So how do we phenotype stomatal density? So we're very lucky in Andrew's lab. We have access to free optical profilometers that allow us to use confocal microscopy to rapidly scan the surface of frozen leaf samples, which allow us to generate these 3D renderings here where you can easily see stomata and the associated guard cells. These 3D renderings are transferred to our collaborators at the University of Wisconsin, Edgar Spalding and Nathan Miller, who are developing machine learning algorithms to automatically count the number of stomata we have in these fields of view. The results files are transferred back to us automatically for analysis, and this all happens via a Cyverse data transfer pipeline. So to Nathan's machine learning counting algorithm, so what we need to be able to do is count the number of stomata that are in these patch and identify the density within each patch. So we need to be able to extract features such as stomatal width that distinguish things that are stomata from things that aren't stomata. So what Nathan does is he models a square image patch like that, overlays disks on top of that patch from which we can get a periodic signal for each disk, which is based on the grayscale. He uses a fast Fourier transformation for each of those periodic signals, so that when we stack all those FFTs into one, we can distinguish clearly between a stomata, get a signal that clearly distinguishes a stomata from a non-stomata. And what was really important, and one of the barriers to achieving this algorithm was how the stomata are not always perfectly aligned at an angle that's consistent between every time we do a scan. Sometimes it's slightly bent, or sometimes the stomata themselves are slightly at a different angle, like you see here and there. So what's really nice with this algorithm, it doesn't matter the rotation of the stomata, we always get the same signal. So we're able to identify stomata rapidly and accurately. And then Nathan can model several features within a patch and extract many features, and then use that to train a pattern neural network, so that as soon as our data, which is transferred to him automatically on a daily basis, is received, it's run through that algorithm into post-processing, and then we're able to get the data back transferred the same day. And as you can see, this is sorghum data here. We have a high level of agreement between Nathan's automatic counts and our manual counts. And actually, with a later version of his algorithm, we're getting correlation coefficients of above 0.9. So we're really getting there really nicely. And the key question for us, like I said earlier, was trying to identify the genes that are underlying the variation in stomatal density, which we see a lot of variation. So we just got a big bulk of this data back now for a project that Kevin's working on maize, where he's phenotyped 200 lines from a recombinant inbred line population in maize, and we were able to do QTL mapping with that data. And one of the QTL that we picked up just last week co-localizes with a known stomatal development pathway gene, which gives us further validation that this Nathan's algorithm is working really well. And we're also picking up novel QTL that potentially represent important candidate genes that are not known yet. So we're really excited with how Nathan's algorithm is allowing us to use machine learning to answer key biological questions regarding the genetic architecture of stomatal density and patterning in C4 species. We're looking forward to use that in the future with various other projects that we're working with. And I think I've probably got time to answer a few questions if you have any. Yeah.
You said the identification of the, sorry. The identification of the, you're doing stomatal density, but with your identification algorithm, can you tell whether the stomata are open or closed?
No.
Right, because I mean,
So
you're
it's
going to
the
have an
way-
environmental,
Yes, the
Yeah.
way we sample, the way we sample is we go into the field in the morning, we harvest leaves, we take them back to the lab, and then flash freeze them.
Okay.
So the stomata shut down immediately.
Yeah, I can't get-
One thing we're trying to work on is exciting stomatal opening
Right.
with we're trying with blue light, and then we're going to flash freeze them instantly and see if the stomata stay open. But that's a question that we're hoping to answer. I mean, we don't want to just use this algorithm for doing density. We want to look at patterning, width, depth also, and that's stuff that we are exploring too. And we're getting preliminary results for that stuff coming up
right,
soon.
right, cool.
Yeah.
Sorry, I question. What's the instrument that you use for the imaging, and is this something like that could be? I mean, is it possible to get some approximation of this into the field? I guess would be.
No, it's a pretty heavy-duty microscope that you wouldn't be able to transport into the field. It's not really been had any. This microscope hasn't really been used for any biological applications so far. It's mainly used in industry to measure the surface of engines, for example. Volkswagen use them a lot. Yeah. They're pretty heavy-duty. You wouldn't be able to get them in the field. Okay.